Hello, everyone. My name is Adarsh. I am presenting a research article titled as Quantification of the Internal Resistance Distribution of Microbial Cells. This article is published in 2008 in the Journal of Environmental Science and Technology, which is part of ACS publication. The outline of the presentation is Introduction, Research Problem and Objective, Materials and Methodology, Result and Discussion, and Summary of the article. In the introduction part, let's discuss about the microbial fuel cell. Microbial fuel cells are a bio-electrochemical device has attracted particular interests in environmental and energy fields. It uses microorganisms as electrocatalysts to conduct oxidation, reduction reaction, and convert chemical energy in wastewater into electrical energy. Microbial fuel cells can be mainly classified into single-chamber and dual-chamber microbial fuel cells. According to the structure, the dual-chamber microbial fuel cells contains an anode and a cathode chamber separated by a membrane as electrolyte bridge, while only anode chamber was included in the single-chamber microbial fuel cells. Membrane is an important component of the microbial fuel cells, which physically divides but chemically and ionically connects the anode and cathode chamber, significantly influencing overall performance of the microbial fuel cells. The core components of microbial fuel cells are anode, where microorganisms transfer electrons; cathode, where oxygen or other electron acceptors combine with electrons; and protons. Proton exchange membrane allows proton flow between anode and cathode. External circuit carries the electron flow from the anode to the cathode, creating an electric current. This slide gives brief about the internal resistance of microbial fuel cells. Internal resistance refers to the resistance encountered by the current as it flows through various components of the microbial fuel cell system. It includes resistances from the anode, cathode, electrolyte, membrane, and the microbial biofilm. The key factors which influence the internal resistance are electrode resistance, the conductive properties of the anode and cathode materials; electrolyte resistance, ionic conductivity of the medium separating electrodes; membrane resistance, resistance from the proton exchange membrane; biofilm resistance, resistance due to microbial growth on the electrodes, influencing electron transfer efficiency; impact on microbial fuel cells performance. High internal resistance reduces power output, efficiency, and overall performance of the microbial fuel cells. Identifying and minimizing internal resistance is key to enhancing efficiency and optimizing design. In this paper, a new method was developed to calculate the contribution of the anode, cathode, electrolyte, and membrane to internal resistance. Experiments were conducted to investigate the limiting factors in single-chamber microbial fuel cells by varying the anode surface areas, cathode surface areas, and phosphate buffer concentrations. The method was also successfully implemented to calculate the distribution of internal resistance of two-chamber microbial fuel cells using the data reported in a previous study. The research problem of this article is microbial fuel cells have performance limitations due to internal resistance, but its distribution across various components is not well understood. The objective of this article is to quantify the internal resistance distribution of microbial fuel cells and analyze how it affects their efficiency. Figure 2 shows the A schematic and B laboratory scale prototype of single-chamber microbial fuel cells used to generate electricity from wastewater. Let's see the microbial fuel cells construction and operation. Single-chamber membrane-free microbial fuel cells, 12 milliliter liquid volume, 1.7 centimeter electrode spacing, were constructed. Used to investigate the power density at various anode areas. 1 to 7 centimeter square, non-wet proofing, type A ETEC, using 7 centimeter square carbon cloth cathodes. In addition, a double cathode reactor setup was arranged wherein the 1 centimeter square anode was placed equidistant between two 7 centimeter square cathodes in order to further investigate the maximum power density generated using this carbon fiber cloth. The cathode has a platinum loading of 0.5 mg per centimeter square point-coated with a PTFE layer to prevent water loss. The microbial fuel cells were inoculated with a mixed bacterial culture from the anode of a single-chamber microbial fuel cells, which was originally inoculated with domestic wastewater and has been operated for about 1 year using acetate. All microbial fuel cells were operated in batch mode using nutrient medium amended with acetate, 30 millimolar. The electrolyte replaced when the voltage decreased to less than 30 millivolt. The system was operating under steady conditions when voltage output was reproducible after refilling reactor with medium at least twice. Repeatable power output obtained in about 50-hour, 3 batches, at a fixed external resistance of 1,000 ohm. Resistance was gradually decreased from 1,000 to 60 ohm for polarization curve generation. At each resistance, microbial fuel cells were run for at least 2 batches for reproducible voltage output. The 10 data points around the maximum voltage at each batch were averaged and used to make the polarization curve. 2 to 3 weeks, it means 15 to 20 batches, of operation was sufficient to finish one polarization curve. Figure 3 shows the polarization curve of a microbial fuel cells, shows the 3 regions of polarization over potentials. A polarization curve is commonly used to study the performance of a fuel cell and the various current-related losses, and includes 3 regions. 1. The cell voltage drops rapidly and non-linearly in the activation region. 2. Followed by a slow and near-linear drop in the ohmic region. 3. The continuous current increase may reflect the concentration polarization region or a condition where mass transport of reactants becomes the limiting factor. In this region, the voltage drops rapidly and non-linearly with the current increase. Figure 4 shows the comparison of observed at voltage and B and nodic power density points and the calculated results lines as a function of current density in microbial fuel cells with different anode areas, cathode areas, 7 and 14 centimeters square, and phosphate buffer concentrations, 50, 100, and 200 millimolar. To explore the power density generated by the mixed culture using carbon cloth electrodes and the relationship between the anode and cathode surface areas, the cross-sectional areas were systematically decreased from 7 to 3.6, 2, and 1 centimeter square using single or double cathode arrangements. Decreasing the anode surface area relative to the cathode surface area increased the power density. A maximum power density of 1050 milliwatt per meter square was obtained at a current density of 0.39 milliampere per centimeter square in microbial fuel cells using equally sized electrodes, 7 centimeters square. This result is comparable to a previous study using the same electrode materials and similar solution chemistry. Reducing the anode surface area from 7 to 1 centimeter square resulted in a 430% increase in anodic power density, 5,570 milliwatt per meter square. A power density of 6,860 milliwatt per meter square was achieved when double cathodes, 14 centimeter square, were used, corresponding to an anodic current density of 2.62 milliampere per centimeter square. The power density of the reactor was also greatly affected by the concentration of the phosphate buffer. As demonstrated in Figure 2, the measured maximum power density was 2,680 milliwatt per meter square for microbial fuel cells with 50 millimolar phosphate buffer. The maximum power density increased by 49% to 4,000 milliwatt per meter square when the buffer concentration increased to 100 millimolar. Further increasing the buffer concentration to 200 millimolar resulted in an additional 39% increase in power density. Table 4 shows the internal resistance distribution and anodic maximum power density of microbial fuel cells with different electrode areas and phosphate buffer concentrations. 6 microbial fuel cells with varying electrode areas and phosphate buffer concentrations. Here's a detailed interpretation. For all tested microbial fuel cells, the electrolyte resistance accounted for 36 to 78% of the total resistance, while the resistance of the anode ranged from 2 to 35%. For the microbial fuel cells with equally sized anodes and cathodes, 7 centimeters square, and 200 millimolar phosphate buffer, The anode only accounted for 5.4% of the internal resistance, while the cathode and the electrolyte each contributed 47.3%. Increasing the phosphate buffer concentration from 50 to 200 millimolar greatly decreased the total internal resistance from 208 ohm for microbial fuel cells number 2 to 85.9 ohm for microbial fuel cells number 1, whereas the contribution of electrolyte to the internal resistance decreased from 78.2 to 47.3%. The decrease of anode area from 7 square centimeters to 1 square centimeter only slightly increased the internal resistance to 235.7 ohms for microbial fuel cells number 3, because the electrolyte 50 millimolar phosphate buffer was still limiting the performance of the microbial fuel cells. The microbial fuel cells number 4, with a 1 centimeter square anode, 7 centimeter square cathode, and 200 millimolar phosphate buffer resulted in a more balanced internal resistance distribution. Doubling the surface area of the cathode resulted in an 18% decrease in internal resistance from 113.6 ohm to 93.3 ohm for microbial fuel cells number 5. Till last slide we discussed about single-chamber membraneless microbial fuel cells. This slide shows the dual-chamber microbial fuel cells construction and obtained experimental results. The figure 5 shows the microbial fuel cells, consisting of an anaerobic chamber, anode, right, an aerobic chamber, cathode left, connected by a glass bridge containing a Nafion membrane. The cathode chamber is sparged with air to provide dissolved oxygen to the cathode, and figure 6 shows the power, A, and voltage, B, generated as a function of current at different PM areas under different electron acceptors of either air, oxygen, or ferrocyanide at the cathode. Anode, carbon, and cathode, carbon with point, surface area keeping constant around 22.5 centimeters square. For the present article, before we discussed about the single-chamber microbial fuel cells with varying the anode, cathode area, and also phosphate buffer concentration, but for dual-chamber microbial fuel cells, they kept anode and cathode area constant at around 22.5 centimeters square with variation of proton exchange membrane area. Figure 7 shows the comparison of A, the observed voltage, and B, anodic power density, points, and the calculated results, lines, of microbial fuel cells with different PM areas, 3.5 centimeters square, 6.2 centimeters square, and 30.6 centimeters square. The open marks and dashed lines are the results for air cathode microbial fuel cells, denoted air, and solid marks and solid lines are the results for microbial fuel cells using ferrocyanide as the catholyte, denoted Fe. The obtained results are discussed in Table 2. Table 2 shows the calculated internal resistance distribution and maximum power of two-chamber microbial fuel cells with different PM areas. The calculated EB, 0.739 volt, of microbial fuel cells using ferrocyanide was higher than the 0.652 volt of air cathode microbial fuel cells. The relatively lower internal resistances of microbial fuel cells using ferrocyanide also contributed to improved power generation. The major, 38 to 86%, contribution of the PM to the total internal resistance indicates that the PM may limit the performance of two-chamber microbial fuel cells. Therefore, the role of the PM needs to be carefully evaluated and considered in the design and operation of microbial fuel cells. Figure 8 shows that the calculated power agrees well with the experimental data. The calculated internal resistance distribution of the two-chamber microbial fuel cells is listed in Table 3. In Table 3, we can see the comparison of the calculated parameters of single-chamber and two-chamber microbial fuel cells. The calculated electrolyte resistances for the two-chamber microbial fuel cells is 33.6 ohm, which is lower than that of single-chamber microbial fuel cells, 40.7 ohm, possibly due to differences in reactor dimensions and electrolyte chemistry. The EB value, 0.5004 volt, of the single-chamber air cathode microbial fuel cells was lower than the 0.652 volt of two-chamber microbial fuel cells, which may have resulted from the higher oxygen crossover in the single-chamber system. However, the much lower internal resistances of single-chamber microbial fuel cells make it possible to produce more than a 10-fold increase in power density. The single-chamber microbial fuel cells and the two-chamber air cathode microbial fuel cells use platinum as the catalyst and oxygen as the electron acceptor and the cathodic ASR of the two-chamber system was 8 times higher than that of the single-chamber system, indicating direct air is much better than dissolved oxygen in supplying oxygen to the catalytic sites. The difference in supplying oxygen might be the result from the much higher oxygen concentration and mass transfer rate in air than those in water. Although the microorganisms and exopolymeric substance on the cathode of a single-chamber microbial fuel cells might increase the resistances of the cathode. The cathode resistance of the single-chamber microbial fuel cells was less than 1/8 of the cathode or membrane resistance of the two-chamber microbial fuel cells, indicating the resistance caused by microbes and EPS was not significant. The high membrane resistance, 2,729 ohm centimeter square, which is not present in membrane-less single-chamber microbial fuel cells, it is another important factor that caused the poor performance of the two-chamber system. The inclusion of a PM, such as Naphthalene-117, needs to be carefully considered due to its high area-specific resistance, which could contribute 38 to 86% of the total internal resistances of two-chamber microbial fuel cells. The summary of the article is: the developed method allows for the evaluation of limiting factors in microbial fuel cells and predicts power generation. Applicable to various microbial fuel cell systems, including single-chamber and two-chamber designs. Parameters such as Ra, anode resistance, Rc, cathode resistance, Rm, membrane resistance, and Re, electrolyte resistance, directly correlate with microbial fuel cells components: anode, cathode, membrane, and electrolyte. These parameters help evaluate the performance of individual components and the overall system. Comparing these parameters across different microbial fuel cells types provides insights into reactor design, material selection, and electrode development. The method highlights that electrolyte, membrane, and cathode resistances are the primary contributors to internal resistance. Enhanced microbial fuel cells performance is achievable through improved cathodes, reduced internal resistance, and optimized electrode spacing, buffer concentration, and membrane selection. A notable anodic power density of 6,860 milliwatt per meter square was achieved using a single-chamber air cathode microbial fuel cells, demonstrating the potential of microbial fuel cells technology. Thank you for listening.